Hello everyone, in this lecture, we will look at the amazing process of DNA replication. Okay, here is a brain teaser from our previous lecture. According to the Chargaff's rule, the ratios of A to T and C to G should be always at 1.00. But you don't really see these perfect ratios from this table. Why is it so? Now there are two reasons. Number one, you have to consider that these data were generated by experiments, which means you tend to get some experimental errors. While number two is about sample size. For human beings, our genome size is about 3 billion base pairs, but for other species, they are much smaller. So for example, E. coli is having a genome size of 4 million base pairs. So as the genome size or as the sample size is getting smaller, there is a higher likelihood that you'll get a distorted value for these ratios. Okay, let's come back to our topic today. Every day, we have billions of cells undergoing cell division, and that means the whole genome, which consists of 3 billion base pairs, is replicated billions, if not trillions of times a day. Just think about this. It is an extremely challenging task because if anything goes wrong, you will get mutations. But amazingly, our body can do this with only one error in every 10 billion base pairs, which is translated to only 0.33 mutation per genome replication. Just how amazing it is. So, how is DNA replicated in our body? This is a topic we want to study today. In the first part, we will look at the semi-conservative theory for replication and the experiment that prove it. And then we'll look at the structure and function of DNA polymerase, which is the most important enzyme in DNA replication. And last but not least, we will look at E. coli as a model to understand key steps in DNA replication. Okay, before we proceed, here is a quick revision. First of all, DNA is double-stranded, and the two sugar phosphate backbones are joined together by complementary nucleotide bases. So, what are the chemical bonds that hold this structure together? Yes, you have hydrogen bonds in between. And then you must remember that those strands are anti-parallel, which means they are running in the opposite direction. So one strand will run from 5' to 3', while the complementary strand will run from 3' to 5'. And again, one strand is the complement of the other. So showing here is a strand of DNA. Can you please predict the sequence of the complement strand? Well, here you will see the answer. It is CGGACCT. Always remember, you have to read the sequence from 5' towards 3'. Back to the 1950s, scientists were very excited with the discovery of DNA structure, but they did not know how exactly DNA replicate themselves. But logically, however, there are three possible models of DNA replication. In the first model, the parental strand would direct the synthesis of two daughter DNA molecules in such a way that the double helix are conserved. So this is called a conservative model. Then you have the semi-conservative model. In this model, two parental strands will separate and act as template to make a copy of itself. In this way, the daughter molecules will have an old strand in blue and a new strand in red. And in the third model, you have the dispersive model. In this model, materials within the parental strand are distributed randomly in between two DNA daughter molecules. So which one is true and how do you prove it? Well, it turned out that the semi-conservative model is true. Basically, what you have is the parental strand of DNA that will separate into two strands. Then each of these strands will refer as a template to produce one new strand of DNA. So this model was proven by the experiment carried out by Michelson and Storr. So this is their experiment. Firstly, they labeled the DNA of E. coli with a heavy radioisotope, which is 15N. Then they transferred the culture of E. coli into the medium containing the normal nitrogen, which is 14N. So if the model of semi-conservative is true, you expect one strand to be 15N, while another strand is labeled with the 14N nitrogen. So that turned out to be true. As for the second generation daughter molecules, you have similar observation that the two strands will again separate and a new strand of 14N labeled DNA is added. But here is a very important question. Practically, how did them distinguish in between the 15N labeled and the 14N labeled DNA molecule? Well, they use centrifugation because the denser 15N labeled DNA will move further in the tube. So for the first generation, they got something in between, in the middle, while for the second generation, they got a mixture of 14N labeled and the 14.5N labeled DNA. Now tell me, what will happen if the conservative model is true? What bands pattern will you see? Well, you always get either 14N and 15N. And to be more accurate, you'll get 14N for the parental molecule, while the rest, all the daughter molecules, will be in 14N. How about if the dispersive model is true? What will you get? Well, in this case, you will always get unpredictable results in between 14N and 15N because this process, dispersive model itself is totally random. Okay, showing here is an overview of the DNA replication process. First of all, the double strand will unwind themselves, and this structure is known as a replication fork because it looks like a two-pronged fork. And then a new strand is formed by the addition of complementary bases such as A to T and C to G, and as a result, two molecules are made, and each one has one new strand and one old DNA strand. And one point to remember is that DNA strands can only be extended in one direction, which is from 5' towards 3'. And the mechanism of replication is actually very similar in both prokaryotes and eukaryotes. And now we'll look at the second part of this lecture, which is regarding DNA polymerases, the most important enzyme in DNA replication. As the name implies, DNA polymerases extend DNA to make a polymer, so it is called a polymerase, and it does so by matching the existing DNA bases with complementary nucleotide bases such as A with T and C with G. And there are several types of DNA polymerases, but they do share something in common. For example, all DNA polymerases can only add nucleotide towards the 3' end of an existing strand, meaning that the synthesis of DNA will not start without a small chunk of RNA, which is known as a primer. So it is labeled as the yellow chunk in this diagram. And all DNA synthesis will happen from the 5' towards 3' direction. But here is one important question. How do you get that yellow piece of RNA primer? Well, you will need the help from another enzyme, which is called RNA polymerase, to synthesize that RNA primer. One unique thing about DNA polymerase is that they have a very low error rate, and that is important to make sure that the genetic information is passed down faithfully and accurately to the next generation. And if you look at the structure of DNA polymerase, it looks very much like your hand. It has got fingers, it has got a palm and a thumb. So in the center, you have the active site by which the DNA double strands can fix right in it. So what are the strategies to ensure a high fidelity replication? First of all, the active site helps to select the correct nucleotide to be added. So that happens right in the P site. And then also in the E site, you have this 3' to 5' exonuclease proofreading activity. So imagine when there is a mistake, you will see that the DNA synthesis process is slowed down due to that distortion in hydrogen bonding. And as a result, the DNA strand will be positioned into this compartment E and be edited with the correct nucleotide base. So you can say that the proofreading activity is almost like a backspace button, where you can hit and erase whatever is wrong. And last but not least, you have the third mechanism, which is a mismatch repair. So the first two strategies are the built-in functions of DNA polymerase, while the last strategy is associated with the cell, by which you have several enzymes such as endonucleases and exonucleases to help correcting the mutation, but you don't really have to go into details with the last strategy. So overall, the error rate is only 10 to the power of -9, or one error in every 10 billion bases. That is very, very low. And now let's look at E. coli as an example. It has 3 DNA polymerases, while for human being, we have up to 14 DNA polymerases. So the function of DNA polymerase 1 is to act on the lagging strand to replace RNA primers with DNA. Now remember, without an RNA primer, there will be no DNA synthesis. And therefore, how do we replace the RNA primer? First of all, this RNA primer will be degraded by RNase H and then followed by DNA synthesis by DNA polymerase 1. As for DNA polymerase 2, it has important function in DNA repair processes. As for DNA polymerase 3, that is the major or the main replication enzyme. So just for information, all these 3 DNA polymerases have the 3' to 5' exonuclease activity, which in other words, they have the proofreading activity. Now we'll look at the action of DNA polymerases. So in order for DNA polymerase to function, it will require the presence of a template DNA, which will serve as a reference for replication. And you also require the presence of the RNA primer, which will provide a free 3' hydroxyl group so that it will get subsequent DNA synthesis. Not only this, you will need the presence of nucleotides, which are in the form of DNTPs, or deoxyribonucleotide triphosphates. So these are the building blocks for DNA molecule. And last but not least, you also need cofactor metal ions, such as magnesium 2 positive, for this enzyme to function optimally. And in fact, if you tweak the level of Mg2 positive differently, you can affect the error rate of the DNA polymerase. And now we'll look at a bit more details with DNTPs. Basically, you have deoxynucleotides. They are attached with 3 phosphate groups. And these are all the variants you have, which are DATP, DCTP, DGTP, and DTTP. So every time when a nucleotide is incorporated into the DNA molecule, 2 phosphate groups will be cleaved and removed, while the last one will remain in the structure of the DNA. And for information, all DNTPs are high in energy because they have these high energy and hydride bonds. So why do I say it is high in energy? Now if you look at these phosphate groups, they are all charged with many negative charges, right? 1, 2, 3, 4. So they are -4. So you can imagine that the repulsion force must be so big. But what is even stronger is the bond strength. They will keep all these repulsion force in control. And therefore, the removal or the hydrolysis of 2 phosphate groups will release a giant amount of energy so that this process of DNA synthesis is powered sufficiently. Well, don't you realize that these DNTPs look pretty much like the ATP? Well, the answer is very simple. These are the cousins of ATP, and they are almost similar, especially for DATP. It is almost identical, except at carbon number 2 of the ribose sugar, ATP has an OH group, while for DATP it is a hydrogen atom, while for the rest it is almost identical because they all come with the high energy and hydride bonds.